All right, while people are settling down, I just have a quick couple of pictures. I think I need my volume raised up a little bit, Amelia. No, I can't. All right, everyone. You're probably wondering why I have a slide up that calls Sleepless in the Summer Institute. So, over the years, we've done seminars in here, many, many seminars. I'm addressing virtual students too. I'm going to look right at you. And there's always a few people who stay up too late or get up too early. And we're going to show a few pictures because what I don't want, you don't want to be this person, right? There may or may not be a TA in this slideshow, right? So think about this as you're starting to feel sleepy. This is a 2-for-1, this one. So think about this as you're starting to nod off and get sleepy. Think about what this being in my slideshow next year. Think about that, and hopefully that will help keep you awake as you go through this. This student thought he was hiding behind the camera, and that didn't work either. So this was pre-renovation. So I think he's in a coma. We called a doctor on that one. I like this one too. This is the speaker. You can think he's kind of reaching over to say, wake up. Don Kiefer is a great speaker. He would never do this, but just the juxtaposition of his hand and the student was pretty good. So that's in summary, right? I may not be here taking pictures, but my friends up at MIT AV and MIT Video Productions have cameras in here. Right? So you may not see me taking pictures, but we've got cameras. So think about that when you're starting to nod off or get sleepy. Think about these pictures. So, and hopefully it'll keep you awake. All right, over to Scott.
Hello, everybody. So I am fortunate today to have the opportunity to introduce another exceptional speaker. Today we have Professor Ariel Furst from the Chemical Engineering Department here at MIT. Ariel's research crosses the boundaries of material science, microbiology, and chemical engineering to really try to solve some of the most challenging sustainability and clean tech problems we have currently facing the nation, facing the world really as well. So it's fantastic having Ariel here today. Thank you very much for doing this, and welcome her here today.
Thank you. I don't think I needed it, but yeah. Thank you. Okay, so before I get started, I'll tell you two things. One, the hardest questions I've ever gotten in a talk are when I gave a talk in this program 2 years ago. And two, the talk you're going to see today is the same one that I give to CEOs because y'all have the same level of science knowledge that they do. So you should be very proud. So my lab likes to think about how we can use things that evolution has already created to solve problems in mostly sustainability and environmental remediation. The way we do this is by looking to nature. So there are going to be a couple quizzes early on just to help keep you from falling asleep. So basically, since humans have been inventing things, we've been looking to nature for inspiration. So if you look at the top, we see a suit of armor. It's from the 1500s, and it was inspired by the scales of the pangolin. Does anybody know what the black and white picture is? You can just yell it out.
It's shark skin on a swimsuit.
Yes, it is synthetic swimsuit material that mimics shark skin that was developed for the 2010 Olympics and has since been banned because it makes swimmers too fast. And you can see the shark skin in the blue next to it. Now, how about the red? Yes. And how many of you knew that Velcro was actually inspired by the interactions between the coatings on seeds and animal fur? That's awesome. Usually I get no one. So even things that we think of as solely in the built environment, solely human-derived, are really inspired by nature. And so that's where we develop a lot of our technologies. Now we're going to have another quiz. When we think about reactions in chemistry, what do we think are some of the most important ones for supporting life on Earth? You can just yell it out. Excellent. I think I actually heard them all. So we have photosynthesis, right? Respiration, nitrogen fixation, and then fermentation, which is what happens when things that normally respire oxygen don't have access to oxygen, right? So these are critical for supporting pretty much all life on Earth. Photosynthesis is done by plants and algae to take carbon dioxide and sunlight and basically produce oxygen. Nitrogen fixation takes nitrogen from the atmosphere and converts it into ammonia, which is essential for plant growth. And then we have aerobic respiration, which is how we breathe. And when we don't have access to oxygen, we go through alcohol or lactic acid fermentation. So what do all these processes have in common? What type of chemical reaction are they? This is a hard one. Close. So they're not all exothermic. It really depends on how much energy we put in. But what they all involve is electrons moving around, right? So we've all heard of redox reactions, right? Electrons moving from one molecule to another is one type of chemical reaction we can perform. And the way we can actually directly measure that is with electrochemistry. So my lab uses electrochemistry as a tool to both understand these processes in biology and engineer them to make better technologies. So you've probably heard the word biotechnology a lot on campus this summer. What does it actually mean? It's basically a summary of the intersection between biology and chemistry and engineering with the goal of combining molecular biology and microbiology techniques. So we all remember the central dogma of biology, right? So the way we can engineer these systems is by interfering with one of these processes. So DNA is converted to RNA sequences that are then converted to polypeptides, right? Those are just the long protein sequences, but they're not considered proteins until they fold. So when we think about this, we can think about engineering in a couple different ways. So we're going to talk mostly about microbes today, so that's what we're going to focus on. We can either engineer microbes at their genome, or we can stick synthetic pieces of DNA in called a plasmid. These are just short circular pieces of DNA. So that means we're not actually modifying the genome. So if we sequence this microbe and one that has a plasmid in it, they will look the same. The plasmid is separate. And so the way we do genome engineering is by actually cutting up the genetic DNA in an organism and either deleting things or inserting things. I'm sure many of you have heard of CRISPR-Cas technology. Yes, that is what it does. In contrast, plasmid engineering is a way we can just shove some synthetic DNA into a microbe and have it be completely external to the genome. So we're not messing with the fundamental function of the cell, we're just adding functionality. And the reason that it's important to have access to different ways of engineering these systems is because biotechnology is important across many different areas, right? We have things like diagnostics and vaccines. We can convert biomass, which is a fancy word for all the stuff we don't eat, in crops. We want to use that, right? And then we have things like improving animal feed, making our plants and animals healthier, and ensuring that people all over the world have enough to eat and it's nutritious, right? So these are all really important problems. And so what my lab tries to do is to combine biology and engineering to make inexpensive and accessible technologies. So that's what we mean by inclusive engineering. Teslas are a great example of what we consider non-inclusive engineering, right? If 0.1% of 0.1% of 0.1% of the global population can access a clean technology, it's really not going to make a difference, right? And so we want to make things that are easy to use, inexpensive, and accessible. And one of the main areas where we see a problem is the use of enzymes. So enzymes are just proteins that can do chemical reactions, and they're used industrially at a very large scale. Currently, the market for industrial enzymes is about $8.5 billion. They're used across chemical manufacture, consumer product good production, pretty much everything. And that's because microbes are very efficient at making proteins. So the way this process normally works, has anybody here expressed proteins before in cells? A couple of you, okay. So we can take that plasmid DNA. Remember, we can make that. It's small, synthetic, and we can shove it into a cell. Now, when that cell divides, all of those cells will also have that plasmid. So we can grow those cells up in a fermenter, and we can tell those cells to start doing what the plasmid tells them to do, which is usually making this protein. The problem is isolating that protein downstream. So what that usually involves is busting open the cells, purifying the protein, isolating, formulating. It's about 90% of the cost of using enzymes. And the problem is you also destabilize those enzymes when you take them out of their happy little cell compartment and put them into a big batch reactor. So you not only are making something that's very costly, you're also decreasing its stability. That's the wrong direction for industrial processes, right? So we developed a technology in my lab that basically came out of laziness. I never wanted to have to purify a protein again after my postdoc, because it's the most labor-intensive and annoying part, and also the most expensive and energy-intensive. So what we do is we just add a little sequence of amino acids that tell cells to put the protein on the surface. So now we skip all of that downstream stuff. We put a plasmid in, we tell the cells to make that protein. We no longer have to bust those cells open, isolate, and purify that protein. We can just take these cells and use them directly. And what we found is we can even freeze dry these, so basically making them into like astronaut ice cream, and the proteins remain active. And so one of the first applications of this was degradation of pesticides. So organophosphate pesticides are about half of the pesticides used globally. These are the pesticides, and what we notice up here, does anybody know what sarin, soman, and cyclosarin are? You can just yell it out. Yes, they are all chemical warfare agents. So the reason organophosphates are such good pesticides is because they work on insects and on us pretty much like chemical warfare agents. We can see agricultural workers in the background here. These guys are wearing PPE, protective equipment, right? The majority of agricultural workers globally who have to disperse this don't have access even to masks. So there are about 11,000 deaths each year from acute exposure, and now there are hundreds of thousands of kind of secondary diseases associated. So low-dose exposure over many years can lead to things like infertility and higher rates of cancer. And all of this is basically because agricultural workers are not being protected like they should. So the great thing is, nature has engineered a protein called organophosphate hydrolase that basically degrades this compound. It takes those pesticides and cuts them in half to turn them into things that are no longer harmful. The problem is, this protein was discovered around 30 years ago, and people have been trying to make materials out of it ever since, and it's really unstable. So if you try to put it on a polymer or even like on a mask so that people can protect themselves, it just unfolds. And so this is where we thought our technology could be useful. So this work was done by a postdoc in my lab, Amrita Karvalkar, and an undergrad, Rachel Almark, who started with us as a freshman and just finished her master's in chemical engineering here. And we basically just told cells to put this protein on the surface. And what we found was absurdly increased stability. We can heat these things up, we can put them in high salt concentrations, and they work. And we can see this because the degradation product is bright yellow. And so we can monitor this by UV-Vis. We've all heard of UV-Vis. It just looks at absorbance measurements. And so because that's yellow, we can look at where that compound absorbs and get a measurement of it. It's a pretty inexpensive technique, which is great. The problem is, if you look at our error bars, our limit of detection is around 25 micromolar. That's about 10 times the lethal dose. I would argue anything you're detecting, if you can only detect it at 10 times higher than the lethal dose, it's not an especially useful sensor. Hopefully you guys agree. Yeah. So we turned to biology. There's a transcriptional regulator, which is a fancy word for a protein that turns on or off genes, that is naturally controlled by small molecules that look a lot like that degradation product. And so we basically started out by doing what most people do in biology and making things turn green. We use green fluorescent protein to monitor whether or not that small molecule was present. And it's cool, right? If we have the small molecule, which is PNP, it's green. But hopefully you see the problem, which is even without the PNP, it's still a little green, right? That's called leaky expression, which means our cells are making that protein even when they shouldn't be. And so it's not great for sensing, right? So we turned to a different type of cell that is one our lab uses extensively. It's called Shewanella onadensis. Has anybody heard of it before? So it was isolated from pond scum in Lake Oneida, New York. You can see scientists are very clever. Shewanella onadensis, Lake Oneida. And basically what they found was that the levels of solubilized minerals in this pond were fluctuating over the seasons, and they couldn't figure out why. And it turns out that these microbes can actually spit out electrons if there's no oxygen around and generate a current. So in nature, they do this to reduce insoluble metal oxides and free up those metals for other members of their community. If we stick them on an electrode, though, they generate current. And so now, if we put that same transcriptional regulator under control of basically the circuit that allows them to make current, so we've unplugged them, and we're basically plugging them back in, we now get very high specificity, and it's electrochemical. So when we combine these, normally E. coli need oxygen to live, Shewanella need no oxygen in order to generate current, so we can't grow them together, but we've already killed our E. coli, right? So they don't care. So we can put these E. coli in with our Shewanella and get current generation specifically when we have pesticide present. So now we're doing degradation and detection in a single system. And it's quantitative. And if you remember, our limit of detection for UV vis was 25 micromolar. Our limit of detection here is sub 1 micromolar. What that means is we are now 2 orders of magnitude, 100 times lower in concentration for our limit of detection, well below the lethal dose, which is what we want, right? So that's pretty cool. The problem is transcription factors are really hard to identify. So we don't want to have to identify a new transcription factor for each target molecule we want to detect, right? So I have a fantastic grad student in my lab, Alec Aghi, and an amazing postdoc, Tali Doughton, who were like, well, microbes are really good at sensing things in their environment, right? Like that's how they regulate, that's how they turn their metabolism on and off, that's how they turn respiration on and off, that's how they survive. How do they do that? Well, they have these really cool 2-component sensors where they have proteins that are very selective for a target molecule, and when it binds to that target molecule, it turns on or off downstream protein pathways that affect gene expression. It sounds really similar to what we did with our transcriptional regulator, right? We just add a step. And so that's exactly what we've been able to do. We can basically steal these genes from other microbes that do sensing and stick them in our electroactive microbes to generate electrochemical signals for pretty much any target we want. And this is quantitative, so we've been able to do this for phosphate and all of these other targets, which are really important when we think about soil health. And that's fantastic, but we don't always know the genes we want to look for, right? So this was actually a project. So you remember Alec, grad student. Kruthi was a high schooler in our lab for 2 years, and she's starting as an undergrad at MIT this fall. And she designed a program that allows us to basically take a molecule we want to detect, and it searches the Kyoto Encyclopedia of Genes and Genomes, which is basically most of all the microbes that have been sequenced. And it looks for things that should bind to that small molecule and then identifies gene circuits that look like they're sensing circuits. So if you know a small molecule you want to find a sensor for, you type it into this program, and it will find new sensors. This is just the workflow for that, and one of the proof of concepts for this was vinyl chloride. So you all heard about the East Palestine train crash a few years ago in Ohio. The chemical that was released was vinyl chloride. Currently, nobody has been monitoring this, despite the fact that it's highly carcinogenic. So what Kruthi was able to do was to identify 9 new sensing genes for these molecules in this family. This took her about 2 weeks to do with this workflow. We were able to put them in the cell and validate that they work electrochemically. And so now we have this workflow that allows us to pull out pretty much any sensor we want as long as microbes have evolved a sensor for it. And that's cool, but this is what our reactors look like. They're basically jelly jars. They're not expensive, they're about $5, and they have a carbon cloth electrode in them. They're about 100 milliliters. Our problem is, when we talk about environmental sensing, I'm betting it would be pretty hard to convince most farmers to have a bunch of these on their land, or to convince the EPA to send people out there and monitor these, right? So the other important thing we need to think about is the form factor. It's really cool that we can do this with cells, but if nobody can actually use our device, it doesn't matter. And so Alec, my grad student, thought of RFID tags. RFID tags are basically these little antennae that, when they're connected, basically just emit radio frequencies. So you can sense them with anything from a cell phone to an RFID reader. They can be mounted on things like drones or autonomous vehicles, and so you don't need a person to actually do the readout. So how do we actually turn cells into an RFID sensor? The answer is a hole punch. So if we have an RFID tag, the reason it's on all the time is because that whole circuit is connected. If we just take that middle part that is the main point of connection and hole punch it out, it's off. We can then just attach an electrode modified with our cells. And so when the cells are generating current, it turns our RFID tag on. Does that make sense? And so now we've taken this device that really was meant for a lab and made it into something that's as simple as walking in front of the sensor with your cell phone in the right app. And I hope what you've started to gather is we really like to think about platform technologies. So when we were thinking about the sensors, we didn't want to develop a sensor that just worked for one thing, right? What about those proteins? What else can we do with proteins that nature's already evolved by simply just putting them on the surface of cells, which improves their stability? Well, one of the things is rare earth element recovery. I'm sure all of you have heard the word rare earth elements in the news. And the reason that we care so much about them is because they are essential to most of our technologies, to computers, to cell phones, to sonar and radar systems. So both for kind of defense applications as well as our daily lives, we need these guys. The problem is we don't recycle pretty much any of these because it's too expensive and companies don't care. And the problem is the way we extract these right now is with acid leaching. And so we basically dump acid into mining sites and let it sit, and the acid dissolves the rock and rare earth elements come out. Not a very efficient way of doing things, right? And this is what happens when one of those leaching ponds leaks. Basically, you now have strong acid flowing pretty much wherever it wants to go and destroying ecosystems adjacent to mining sites. So we need new ways of doing this. And so this work was done by a postdoc in my lab, Melanie Goot, and an undergrad, Tatum Willem. Tatum was the captain of the crew team here for 4 years and is now a grad student at Stanford. And there are proteins that have already evolved that bind to rare earth elements very tightly. They can bind rare earth elements specifically at parts per billion concentrations, even when there's really high concentrations of other salts. And so that protein is called LAN-modulin, or LAN-M, which you'll see here. And the great thing is this protein is really stable down to about a pH of 1.7. So it's pretty acid stable. So we can still pull those elements out of the rock, and then we can run them through our column to isolate them. And the cool thing is, if we go below 1.7, it doesn't actually destroy the protein, it just unfolds it. So it gives us a very easy lever for recovery. And so what we've been able to do is to basically make a column packing material out of this. So we just put our cells onto glass beads, and it allows us to make a flow column. And what we see here is over multiple days, we can bind and release rare earth elements and we can pull them out at parts per billion concentrations and basically concentrate them. And the impressive thing here is, so the timing, if you look, it's over 10 cycles in 6 days. The timing is not accurate. It binds over gravity flow, so it takes about 10 minutes to bind, not half a day. But the impressive thing is that these proteins remain stable without refrigeration, just on a benchtop, even exposed to acid. And so when we think about the scalability of this tech, we now have removed all the need for downstream processing in order to use proteins, right? So we don't need any special equipment to make these protein materials, and it's less than 10% of the cost of conventional proteins. And so what this means for rare earth element recovery specifically is it costs us about $10 a kilogram to make these dried cells. We can recover about 12 milligrams of lanthanide, one of the rare earth elements, per gram of cells. And so when you do the dimensional analysis, what that means is it costs us about $150 a kilogram of rare earths to recover them. Currently, the market value for these rare earths is around $2,000 a kilogram. So I would argue at that price point, it would be worth it for companies to actually start recycling these materials, which is our goal. So what else can we do with proteins that have already been evolved naturally? So something I found out when we were thinking about plastic recycling is that people in the US throw out about 80 pounds of clothes per year. That is an enormous amount, and that is not what you donate. This is what literally ends up in landfills. And about 60% of all the textile waste in the US is polyester. So it's a good target, right? If we can degrade about 60% of textile waste, we can make a significant impact on the amount of waste produced. And so what we've been able to do is to take a protein that's actually found in our bodies. It takes triglycerides and turns them into glycerol and lipids. And we can take that protein and put it on the surface. And the cool thing here is that it actually requires another protein called a foldase to make sure it folds well. If we put them both on the surface together, that foldase helps that lipase fold. So we can use chaperone proteins with this setup to maintain our low cost of protein expression. And you can see here, this is a chunk of polyester that we've just left with 0.1 weight % of cells overnight. We get about 70% mass degradation. And you can see for both shorter polyesters and longer polyesters, within a week, we get nearly full degradation with 0.1 weight %. So that means to get tons of clothes degraded, you remember these cells cost us about $10 a kilogram to make. It's effectively free. And the fantastic thing is we are not making microplastics. We're actually going down, sorry, I must have deleted that slide. We're actually taking it down to the monomers to tetramers, so 1 unit to 4 units, which means we can then collect those and use them to make new plastic. Currently, about 80% of plastic feedstocks are made from fossil fuels, so even just recycling textiles would significantly decrease the amount of fossil fuels we need for the polymer industry. So these are just some of the things that we've thought of, but my goal here was also to just get you all thinking about what proteins might already exist that you could use for sustainability challenges and things like that by just simply looking at what's already been evolved. So now we're gonna flip the script a little and talk about how we can use whole microbes for different applications. So proteins are great, but often they don't work very well alone. So how can we take these entire microbe systems and use them? And I want to say, so I'm the co-founder of a startup based on this. So a lot of the agricultural work, we are trying to replace agrochemicals, so fertilizers and pesticides, with microbes. So I don't think I have to argue this here, but microbes are pretty much essential for all life on Earth, right? What's the problem with that? Well, we can't formulate them. So this is data from living biotherapeutics. That's a fancy word for microbes that would be FDA approved as medicines. Over 60 different microbes have gone to preclinical trials. Less than 25 make it to phase 1. For phase 2, you need at least a semi-final formulation. So you move from, in phase 1, they usually make people drink these in their native media, which is pretty gnarly, to phase 2, where they have to be dried. Very few actually can make it to phase 2. Phase 3, only 5 have made it, and none are currently FDA approved. And this is really because of our limited ability to actually make these microbes at scale. Most microbes in the environment that we would want to use are delicate. They like living with their natural communities that help feed them and protect them. And when we pull them out and try to grow them in isolation, they say no. So what my lab has developed are these self-assembling suits of armor. We like to think of them as kind of like Iron Man suits. And they form in 2 dimensions on the surface of a microbe and protect them against pretty much everything we can throw at them. So these suits of armor are made of polyphenols. Has anybody heard of polyphenols before? A couple of people. Things like tannins in wine and coffee, green tea extract, those are all polyphenols. So we already know that they're beneficial to us. Many of them are antioxidants. And we can mix those with a cation, just a positively charged metal ion. We can use things that are earth abundant, like iron or manganese, that are essential minerals for us. And we can buy all these in food grade. So these are basically safe as is. We don't have to do anything to make these safe for use in humans. And the cool thing here is they form really rapidly. So if you watch the video, that blue color is the coating forming on microbes. It forms pretty much instantaneously. And one of my favorite things is based on the metal and polyphenol you use, you get different ligand to metal charge transfers, and you get these really pretty colors. So based on what you use, you can both change the color but also change the physical properties. And so we can image these, and we can show that these form really tight structures around a full microbe, right? We don't want these if they're only forming patches or not coating every microbe in our mixture, right? So what we see on the left is TEM imaging. And so on the very far left, that's just an unstained, uncoated microbe. It looks fuzzy, right? The metals in our coating can actually serve as a stain and allow us to image, and we can see that those metals are fully around the microbe and even form on these subcellular units. So we've seen that we can form them on flagella and other components of microbes that we want to protect. And if we stain those coatings, you can kind of see here, it's a little hard to see on the screen, apologies, but every microbe in this mixture is fully coated, right? So this is a scalable process for industrial applications. So the next thing we had to do was figure out what stresses it actually protected against. So my students got to come up with a bunch of different tests on how to kill microbes in different ways. This is what they came up with. If anybody has additional stressors that you can think of, would love to hear about them after the talk, because we are always looking for new things to try. We can put these in acid, we can put these in base, we can put these in high salt, in organic solvents. We can mechanically stress them. We can put hydrogen peroxide in them. We can expose them to humidity and temperatures that can cook an egg, and they survive. And so now that we have a way to make microbes at scale, what can we do with them? So in agriculture, I mentioned already, we can think about using microbes as replacements for fertilizers. We can also think about enhanced weathering. This is a fancy word for microbes that can basically pull CO2 out of the air and turn it into limestone. Currently in agriculture, to adjust the pH of soil, they truck in limestone. So they mine limestone, put it in a truck, drive it hundreds of miles, and dump it. We can just take dried microbes and have them do it directly from direct air capture. Human health, if we can take microbial vaccines and protect them without requiring cold chain, we can significantly increase accessibility, right? And a lot of the microbes that we want to use for like gut health are anaerobes. They don't like oxygen, and we can protect them against oxygen exposure, so it decreases the cost of their production significantly. When we think about green chemical production, when we want to use microbes that are non-model organisms, so things that haven't been evolved to be very stable in a lab environment, it's really hard. And so a lot of the microbes that have functions like the ability to produce sustainable aviation fuels or sustainable plastics, we can't actually use them. And so these coatings allow us to protect them in a way where we can use them now for scaled production. And finally, when we think about how we can use these in our everyday lives, we can use these for plastic recycling. I already showed you one example of how we can use protein for that. We can think about these for green mining, so removing the need for acid leaching to pull things out of the earth. We can improve recycling so that we can actually recycle a lot of the metals in e-waste that currently just go to landfills. And we'll talk about how we can use these for building materials, which is basically doing direct air capture for self-healing cement. So you've probably noticed we think a lot about cost and scalability from the beginning of a project, right? We don't really want to go down the road of developing something that takes 10 years if it's never going to be commercially viable. So at scale, we know we can produce microbes for about $10 a kilogram. We get around 10 to the 6th to 10 to the 7th more live cells per batch with our process than with conventional growth. So you're basically getting millions to billions more live cells per batch. Our coatings are all generally recognized as safe by the FDA, and we can get them in food-grade quantities. So this means that we minimize the amount of regulatory barriers. And for, say, a year of probiotics, the incremental cost of adding our coatings is 25 cents for the full year, to give you some idea of scale. So what can we do with these? So has anybody heard of the BCG vaccine? One person, excellent. It's one of the few microbial vaccines, and it's a vaccine against tuberculosis. It has been known for about 100 years. It's basically cow tuberculosis, so it is similar enough to human tuberculosis to be useful for our immune systems to train on without being deadly enough to actually kill people if you're injected with it. The problem is, most places in the world where tuberculosis is prevalent don't have consistent access to cold storage. So it's really hard to get these vaccines where they need to go. We can now protect the BCG vaccine for 3 weeks at 50 degrees Celsius. That's about 130 degrees Fahrenheit. So with that, we can now transport these vaccines and store them without requiring consistent electricity access or refrigeration. So how about protection of microbes for agriculture? So many microbes can do nitrogen fixation. They can take nitrogen from the atmosphere and turn it into ammonia, basically the Haber-Bosch process, what we do synthetically. The problem is many of these microbes are temperature sensitive. So if they're stored above 25 degrees Celsius, so ambient temperature, room temperature, they all die. Which means the only people that can use microbial fertilizers are wealthy enough farms that can grow the microbes on site. We've been able to protect these against storage at 130 Celsius, or 130 Fahrenheit, 50 degrees Celsius, for 6 months. So now you can store these in whatever shed you have, wherever you keep your ammonia fertilizers, without having to have refrigeration. And what we find is that our stressed cells actually improve germination even more than using fresh cells. So if we compare these to current regenerative agriculture, we beat it by about 150%. Imagine if using something cheap and easy boosted all food production by 150% globally. And that's really what our goal here is. Because you don't have to distribute these multiple times a season, you save cost on labor. Because you can specifically target microbes that secrete pesticides to where they need to go, you save money and environmental damage by not distributing pesticides broadly. We don't have to store hazardous material, so ammonia is flammable, so it's really hard to store long-term for use as fertilizer. And we don't have to use cold chain storage. So we've saved farmers about 50 to 70% over chemical fertilizer costs, while also boosting their germination rates. So the last application I'm going to talk about is one that I think is very cool, which is basically taking microbes that normally make things like stalactites or coral reefs and sticking them in cement. And so these guys in nature can take CO2 from the atmosphere. These are the same ones we think about for enhanced soil weathering. Stick those CO2 molecules onto calcium or magnesium and basically make limestone. And so this would allow you to do direct air capture, basically for free, while also decreasing your building costs, right? Because you no longer have to send someone out to do maintenance and repair cracks. So these microbes can make these very large calcium carbonate particles. So this is just an image of the particles after about 8 hours. And you can see the microbes in green. And what we want to look at here is the fact that these particles are forming outside the cells, which is exactly what we want, right? If you're growing these inside the cells, eventually you'll reach a maximum where the cell dies. And so you have an upper limit to how big you can make these. The other really cool thing we found is you can control the crystallinity of your particles by changing the microbe you use or the counterion. So if we use calcium acetate versus calcium chloride, we change the size of the particles. These dark spots are just the particles. You can see much larger ones here. And if we just change these strains, these are all soil-derived strains, so we haven't engineered them at all. You can change the crystallinity. So these are calcite and these are valerite. And so if you want specific kind of tensile strengths from your construction materials, you can achieve that by just swapping out the microbe you use. And so what we all actually care about is how does this work with cement? And so this was actually a project for a group of high school students in my lab last summer. They mixed in different dry weights of microbes directly into cement mix. And so those pictures on the left are 10 centimeter by 10 centimeter pucks. They then took a hammer and a nail to them. So that hole in the middle is from a nail. And if we just kept them in a humid environment, after a month, we got about 35 to 40% crack healing. And you can see that if you look from day 0 to day 26, you can see the crack is filling in the middle from the bottom up. And we can vary that and actually speed up the processing by just increasing our weight percent. So this is with 0.1 weight percent of microbes to cement mix. We can go up to 2% without negatively impacting the overall compression strength of the cement, which is what we want, right? If we're starting to sacrifice the overall strength of the cement, nobody's going to want to use it. So we need to maintain the original properties, but we can also speed up this process. So I hope what this has shown you is just how to think about, in addition to the proteins, whole cells and what their function is for all of these different applications. And I think I will skip the last story so that we have time for questions. I definitely overloaded this, but so I hope what I've showed you today is when we think about what nature's already evolved, we can significantly improve sustainable technologies that are also cost-effective and easy to use. So that's really our goal and what I want you to kind of think about as you think about developing new technologies. With that, I'm going to thank my lab. We also, I didn't talk about it, but we work with methane producers, so they all dressed up like cows for our group retreat, and they didn't think I was going to exclusively use this picture of them from now on. So yes, I'm excited to take questions, and thank you all for your attention. If you guys can yell loud, we can start. Do you want to start? Yes, if it's coming, yeah.
Hi, I'm over here. Thank you so much for your talk. I just wanted to ask about the nano, armor particles. Does that limit functionality at all? Does it still work the same way?
Yeah, so the armor actually only stays on the microbes when they're in stasis. So really when they're either dried or in nutrient-depleted liquid like water, once they start to grow, they actually break out of the coatings physically and divide and act like normal. So it's really a way to protect them, to get them where they need to go, or to get them through the processing aspects of manufacture to allow us to use them. That's a really good question.
Hello, my name is Lilith, and I was wondering, so since you are developing all these such cool things and you want them to be accessible, have you started putting them out on the market or distributing them for people to use?
Yes, that is a great question. So we have a couple startups from my lab. That is one of the great things about MIT is it really trains students on how to do startups. So all of my startups have students who are working on the projects affiliated with them. Excuse me. And so our goal is to either do a startup with most of our technologies or license them to larger companies that will actually implement them at scale.
Janaya, can you select 2 virtual?
Yes. We're going to have Odina.
Thank you. I'm Obi. So I'm actually in a remote sensing course, so I was really interested in this lecture in particular. But what scalability challenges in like using proteins like lanolin or lanmidolin for like environmental sensing? How would like their stability in extreme conditions open up like new applications in
So the great thing is a lot of our technologies actually kind of do that on their own. So one thing that I didn't discuss specifically, but what we found with our lipase, so the polyester degrader, is it's actually stable up to 95 degrees Celsius, so almost boiling. Sorry. So we can now use these. So the proteins themselves, if we isolate them, are only stable up to about 37 degrees. So we now have a much broader temperature range, right? Which is exactly what we need, because polyesters, those chains, they're like a bowl of spaghetti. Once you start heating it up, they'll start kind of expanding, and it's much easier to chew them up. So we kind of, in a lot of cases, the inherent technology base, which is the surface expression, gives us features that are beneficial, like extra stability. Hopefully that answered the question.
Yeah, thank you.
Okay, and we'll have Sahana.
Hi, yeah, so I was wondering, for all the different applications of microbes, how do you ensure that after the application in the real world, they don't lead to like uncontrolled growth? Or are like the microbes all already engineered to like not replicate?
So that is a great question. All of the microbes I talked about in that second half are not engineered. They're all native strains, and they're all soil isolates. So none of them are pathogenic. All of them are beneficial, and so if they proliferate in the environment, we see that actually as a good thing because it just boosts biodiversity. So especially in agriculture, the use of chemicals as pesticides and fertilizers has basically killed all of the native microbes there. And so a lot of natural soil processes like organic matter degradation and nitrogen fixation are not happening. And so if our microbes stay there long term, that is the best case scenario, right? Because we can mix these in a way that makes them look like a native community. That's a great question.
Hello. My name is Lucy, and I really learned a lot from your presentation, but specifically I was curious about plasmid engineering and how you make the plasmid outside of the cell, how it's inserted into the cell, and then how, since it's outside the genome, is it read any differently?
Yes, those are all fantastic questions. So plasmids basically have a backbone and then a gene insert. And so you can pick your backbone based on which organism you're trying to express it in, what you want to trigger expression, things like that. And then your gene is basically just something you can actually biochemically synthesize, and it's the DNA sequence, when we think about central dogma, that then translates to the protein sequence you want. And so we can literally order those online in whatever sequence we want, and then you can either use, basically there are enzymes you can use to cut these to make ends that will adhere together, or there's a newer technique called Gibson assembly that allows you to just heat them up and then they'll anneal, or the overhangs will basically hybridize and form this circular plasmid. So the way we stick them into cells is also kind of crazy. Basically, we blow holes in cells for a short enough amount of time that it doesn't kill the cell, but is long enough that the plasmid can enter the cell. So we can do that either with heat, so you can either basically stick certain strains of E. coli in a heat block at 42 degrees Celsius for like 30 seconds, or we can electroporate them, which I think is the coolest. You stick them in a cuvette that basically has 2 electrodes in it, and you apply like 100 volts across it, and it blows little holes in it, lets the plasmid in, and then they seal up.
Yeah, thank you. Pass it
down. So I was wondering, for the like suits of armor on them, have you tried exposing the microbes to extreme pressures, such as like vacuums, or is that not really applicable in your use cases?
Yes, that is definitely applicable. So one of the ways we need to formulate microbes that's really hard to do is freeze drying. And the way you freeze dry things is you stick them in liquid nitrogen, and then you put them under low enough pressure to sublime off your water. And so what we do is we basically suck all of the water out of them after it has crystallized. So normally what that does is the ice crystals form in the cell. It tears the cell apart because you now have these pointy crystals where there used to be liquid water. That's step 1. Step 2 is we then apply basically millitor pressure, very, very low pressure, to suck all that water out. And both of those processes basically make these microbes, if they're not protected, look like raisins. Our microbes look like microbes afterwards, and so it does protect against that. Haven't tried high pressure yet, although we've tried osmotic stress.
Can we do 2 more online?
Yep, so we'll go with Huang.
Hi, I kind of have a follow-up question from Satana's question. You said the microbes that you use are local to the community that you apply on. I know all these applications you talked about could be applied anywhere, especially agriculture. Does that mean you have to find a specific microbe for each local community? And wouldn't an external microbe have an invasive effect or disturb their ecosystems?
That is a fantastic question. So there are 2 considerations when we think about especially microbes for ag. One is kind of the general things each microbe can do, and then community-specific things. So we can either go to a location, sequence what's there, and figure out what's there to be beneficial. But then there are also microbes that are pretty universal. So B. subtilis is one of them. This is a microbe that's found in pretty much every environment on Earth, and it can do things like CO2 sequestration and nitrogen fixation. And so we know that that's going to be beneficial in pretty much any environment. So there's kind of 2 different paths we take. One is kind of consortia designer mixtures of microbes for extreme environments, and then the other is just using microbes that we know do these basic functions that are found pretty much everywhere.
Thank you very much. That makes sense.
Okay, and then we'll have Beverly.
Okay, so I was wondering if you had to develop any special methods to test the different extreme conditions you had to test the microbes under.
Yes, that has actually been a really fun part of the project. So what we've been able to do, we bought a spray dryer. So I mentioned freeze drying, right? Spray drying heats up the microbes in liquid basically to hot enough so that they aerosolize, and then it puts hot gas on them to blow off any liquid, and they end up dried. That's how most industrial processing of kind of pharmaceuticals works at scale. So we bought a spray dryer, which is really fun. We've also been able to get samples of things like acid drainage from mines, which we can then stick microbes in and see what grows. We've been able to get contaminated seawater. So we've been able to look at radioactive seawater. Basically, getting samples from collaborators is the other way. So buying weird industrial equipment and then also getting samples from crazy environments.
All right, thank you. At this time, we're going to transition to our virtual t-shirt presenters. So can I have 2 SGI students that have been notified?
And I will be hanging out after, so if you guys have a couple more questions.
That's also for the virtual students. If you guys have questions, make sure to put them in the Q&A. Do you want to stand here?
Hi there, Miss Furst. I'm Ethan.
And I'm Jana.
We're from a course on serious games and artificial intelligence. Thank you so much for your talk.
Yeah, thank you. So personally, I'm also interested in pursuing biotech. So I found it really fascinating that you're using really innovative solutions to ensure accessibility, especially because I feel like that isn't really talked about enough in this field. And so I just appreciate that you really broke down the work that you're doing right now, explaining it in such a concise way, and then expanding it and showing us how it applies to all the ways it connects to our everyday life. So yeah, we really appreciate you for taking the time to talk with us.
Mm-hmm. And as a small thank you for all of the information you've shared with us today, we would like to give you an official BWSI t-shirt.
Right, thank you guys.
Thank you.
Thank you.